What I was trying to do at the end, which probably was the more confusing part, was to show you that the communication from the outside of the cell gets inside the cell. And when that communication, sorry. Oh, is it, mic's not on?
I mean, I'm only getting it in the camera.
It's on. Can you hear me now? You can. It may be because the air conditioner is on as well. So what I was trying to show you was that how stimuli from the outside of a cell get into the cell and how it influences what happens in that cell. That process is very, very important in what we think is going on in the context of MS, not only in terms of other things that other people's work, but also work that I have been involved in, which we're going to continue to try to understand how this process actually happens. Is that a little easier also with the air conditioning off? Okay, good. All right. So now I want to try to show you how the immune system learns to tell apart the difference between our own body and something that's foreign. And the irony of the immune system is actually you have to recognize part of your own body, which are those receptors that we talked about, to also be able to recognize something attached to it that's something foreign. And so one of the major problems the immune system has is how do we control responses against ourselves and yet preserve responses against things that could be harmful. And that's a very, very delicate balancing act. And that balancing act, unfortunately, is lost in about 2 to 5% of all adults at some time point in their lives, which is, if you think about it, 1 in 50 to maybe even in 1 in 20 people who during their lifetime may have some sort of autoimmune disease. That's an incredible number. What's even more of a problem is about 20% of the population have allergies, which is also the immune system going awry, but not causing an autoimmune disease. So that problems of the immune system actually are so common, they're probably, as a generic group, the most common diseases that occur, more even than cancer or heart attacks. So actually, although people don't understand this, understanding the immune system is really critical not only to these diseases, but in fact the immune system plays a role in cancers, transplants, chemotherapies, etc., etc., etc. Diabetes, there are many different areas, head injury, Alzheimer's disease, Parkinson's, etc., etc. So the immune system plays a role in an enormous number of processes that affect us in one way or another. So let's go back then to the immune system. How does the immune system learn to recognize but not recognize ourselves? Well, if we start at those stem cells that we talked about a little earlier, these cells go through, in the thymus, a process of education where they put on that T cell receptor that we talked about, and then they go through a whole process of maturation. Early on, if they don't develop the receptor, they just die. There's a process called apoptosis, which wipes out all sorts of cells. If they don't put on the antigen receptor, they die. Then, if they recognize part of our body too vigorously, they also die. But if they have just a weak recognition of our body, so just enough, like Goldilocks, just enough and no more, they then can leave the thymus gland as a mature cell, which can recognize our body, but in theory doesn't have the possibility of causing an autoimmune disease. So in that way, we accomplish the process of recognizing our own tissue, but not reacting against our own tissue. And there are many different themes that play into this to control the immune system. Now, again, I'm not trying to confuse you, but all I'm showing you is that that receptor that ends up recognizing something, which you see is in little bits and pieces of color, actually is a process arrived at by splicing and dicing many different parts of genes, which are all these different colors. So as you go from the gene all the way through to the protein itself, there's this incredible selection process. So we can select from all of the above which is hundreds of variations, to get the kind of molecule that's tailor-made to recognize something that is part of our cells or something that's part of something else. And that's all I want to point out with that very busy slide. And the same thing is true for antibodies in the way they can recognize things, and T cells in the way they can recognize things. And so we have both of these processes going in parallel. Now, in the thymus gland, as I'd mentioned, there's this process of selecting cells that are not too weak or too strong, not too hot or too cold, and getting rid of cells that either don't recognize anything or recognize our cells too strongly. And as a result, we come out at the other end with these different cells that we call CD4 cells, that are the helper cells of the immune system, and CD8 cells that are the killer cells of the immune system. And so these, again, are mature cells that have this ability to recognize our cells but not respond against our cells. And now, one of the things, and the reason I'm showing you this is primarily because we have the piece of equipment up in the lab which actually has the capacity to identify cells. So the way we do it is, if this is a cell over here on my fist, we take an antibody that recognizes part of that cell and has tagged onto it what we call a fluorochrome. If I shine a laser at it, like I'm shining the laser from this pointer at it, and it hits what's called the fluorochrome, it lights up. And when it lights up on that cell, there's a complex mechanism in that machine which actually can then tell us, for example, here's more CD4, here's more CD8, which are those two different kinds of T cells. It can tell us whether it has neither on them, both of them on it, or them individually. And we use this mechanism, and I'm only showing you this because we use this mechanism to identify specific kinds of cells. Not only can we recognize what's on the surface of the cell, but we can use the same technology to get an antibody into the cell. So we can determine which kind of cell, which flavor of Baskin-Robbins it is, and what's going on inside that cell of any particular pathway that we're interested in. And we can do it all simultaneously so that we can use different antibodies with different colors, and we can do mix and match. In fact, our machine does 6, but there are machines that can do as many as 20 or 30. It becomes extremely complicated because there are only so many colors in the color spectrum, and you have to actually separate them. But we have the ability to label exquisitely individual cells and what they're doing to understand the process of what I'm actually talking about. And we have that capacity upstairs. So going back to the immune cell, normally, as you remember, the dendritic cell picks up a little bit of whatever and instructs these cells. These cells become activated and then come out and can circulate in the body, either against our own tissues in the case of MS, or something that's foreign. And the net result, again, if you remember, is that we have different cells that do different things. Now, what's very important about this is that when the activation process happens here, there's an amplification process so that more and more cells of the same type are made. So if you recognize part 1 of a molecule, you're not only going to have one cell that recognizes it, but you're going to have dozens or hundreds or maybe even thousands of cells that can recognize it. And that amplification allows us to have a very effective process of getting rid of something. But the downside in terms of autoimmunity is if you have a whole lot of cells around that recognize your own tissue, that's bad. So the same process that's good on the other side of the coin can end up being a process that's bad. And what makes it even more difficult is not only do these cells go ahead and activate cells or cause inflammation, but they also turn into what are called memory cells. So even if you, for example, get rid of a virus and these cells settle down and maybe all die by what we call apoptosis, that's programmed cell death, there are cells that are left that remember. So if you remember early on when I told you that after a primary or a first immune response, the second response is more vigorous and quicker, The reason it's more vigorous and quicker is that these cells have already been instructed to remember what was there. So if they're activated, they potentially are more helpful against the virus, but more harmful in terms of causing an autoimmune disease. And in fact, this process by which these cells become activated is a very, very sophisticated molecular process where individual genes get turned on in a sequence. So they don't always get turned on all at once. It's a programmed pattern of behavior that then, for example, here will lead to a cell dividing. And cells, as you can see, have these little different cartoons on them, and they go through a process, not only amplification, but at the end of it, they start putting molecules on their surface which put on the brakes, so that that curve goes up, but that curve also comes down. And that's very important again, because in certain instances, if the reaction has occurred against one's own tissue, we need to be able to rein that in. And there are different ways by which it can be reined in, some of which are inherently there in the surface of the cell that might be responding against our cells. So even that cell could potentially turn itself off under the right circumstances.